Welcome everyone to day 2 of Microbiology Lab. We're working towards lab practical number 1 if you're in my Bio210 class. This video may also be used for my Bio114 class, at least as we're learning some of the basics of how a microbiology lab works. So we're going to continue on. Let's take a look at the syllabus, and then we'll get to continuing day 2. I just want to remind everyone, as well as myself, about what it is we need to cover through day 2. So we're going to go back and make sure that we're complete on all of the subjects and reviewing the readouts of the setups from the subjects on day 1. And then we'll finish day 2 with aseptic technique and finally the negative stain. So those are the items that we need to take care of today. They're on our agenda. And now I'm going to skip to reminding everyone about our environmental cultures that we set up on day 1. And we'll take a look and see what came of our environmental culture setup. If you recall, a plate was labeled on the bottom, and a swab was used after it was wetted. We put this to sample the environment and pick up a sample that we then spread around on the plate. And I decided to make 3 samples for us to look at. One of them was a 20-minute air sample. And the 20-minute air sample was just out here on the desk in the first video. And the air in here is pretty still. I do know that there's some movement from the air conditioning, but surprisingly, the plate has nothing growing on it. Certainly that doesn't mean that the air inside of MicroLab is sterile, but it is nice to know that at least it's not blowing around a bunch of dirt and dust. If we had put this sample outside for 20 minutes, which would be sort of a control by comparison that we could have put out, typically we would see some growth, whether it would be some random mold spores, so you'd have some fungus that could grow in here, maybe some bacteria that are blowing around in the air as well. So this was a little bit anticlimactic, but that doesn't mean that there's nothing on here growing. Remember, we've only been growing these plates for 48 hours in the incubator at 37 degrees Celsius. So I'm going to leave this plate over the weekend, but at room temperature, and we're going to see if more bacteria grow, or some bacteria, or maybe some fungi. And so we'll write that down. Additional incubation at room temperature. That's what we're going to do next for 5 days. So in total, we'll have 7 days worth of possible growth on our plates. But before we incubate these all at room temperature, which may give mold a chance to grow, because they grow better at room temperature and they grow more slowly, let's take a look back at my other samples. They're much more interesting than the empty plate that the room air produced. Here's a plate that's from my left shoe, and it has a whole lot of growth here that we'll take a closer look at, and also the plate from my cell phone. Ew, it grew a lot as well. And so let's take a look at these a little more close up. I'm going to switch over to the document camera, and we'll zoom in on these. I'll place a black background on here with a dark background. Should provide a little bit more contrast. Wow. So, no big surprise in one sense that the bottom of my shoe, which comes in, undoubtedly comes into contact with a whole bunch of dirt and just whatever's around on the ground. Contamination generally is going to fall down due to gravity, And the bottom surfaces of just about anything in an everyday environment is going to be very contaminated. And you can see the vast array of different microorganisms growing here. And now they're not so microscopic. They're actually, every one of these colonies, like this little yellow colony, we would call that like a small or maybe pinpoint type colony, that represents 1 million cells. That's impressive. One cell maybe was on there, an invisible single cell, which then grew into 1 million cells. And that visible colony right there, never mind these massive ones, just that little visible colony may have became visible as early as yesterday, over 24 hours or less. Really what we need to do is think about what it means to make a colony. I'll come back to this because there's so many interesting things to look at. So let's look at bacterial growth for a moment and try to understand it a little bit better. On this slide here, on the first day, I was explaining the term culture, and also I believe that we defined the term colony somewhere. Let's see if we can find that. I'm skipping around for a moment till we see the word colony, right? A colony is a visible number of cells that amounts to the millions of cells. And so let's do a little math exercise, all right? If one bacterium takes 20 minutes to double, that's pretty average. So staph, like that little yellow colony, is probably a staph skin type of bacterium, or E. coli, ordinary gut bacteria, about once every 20 minutes under ideal conditions where they have plenty of food and space to grow and 37 degrees warmth where they can grow their fastest at human body temperature, then that means that they're going to be able to multiply 3 times an hour. So how many doublings would it take to go from one invisible cell all the way up to, let's say, 1 million, okay? Now, think this exercise through in your head. I'm going to go ahead and establish, I'll just use this as a blank area for this purpose. I don't have it pre-prepared, but it shouldn't be that difficult. We'll do it, well, perhaps, why don't we just do this in our heads? So how many would it take to go from one cell, how long would it take to go from one cell to 2 cells? So that's 20 minutes, right? Okay, so then 2 cells double after another 20 minutes and they become 4 cells, okay? The way I want to count this out is let's do it by generations, all right? So one generation gives us 1. Generation 2 gives us 2. Or one generation, we go from 1 to 2, okay? Next generation, you go from 2 to 4. And then 3rd generation, we go to 8, right? So 3 doublings. And then 4th generation, 8 becomes 16. And then 5th generation, we now have 32 bacteria. And then 32 doubles to 64. And then 64 doubles to 128. And then 128 doubles to 256. And then 256 doubles to 512. Maybe some of you are familiar with these if you think of exponents or like doubling. Computer parts are sold this way, right? So 512, that's 9 generations. And then double that and you have 1024. So just over 1,000 bacteria after 10 generations. 11th generation, that would be 2048. And then after that, you would have 4096. And then after that, you would have 8,000. Oh my gosh, what would it be? 8,000. Can't remember the exact number, but let's not try to do all that math in our heads. Let's round it to 8,000. That'll be easy enough. And then we have 16,000. And after it was 8096. I remember it now. Then 16,000, we'll just round it. 32,000, 64,000, 128,000, 256,000, 512,000, now we're at 500,000, and then one more doubling, that makes 20, we're over 1 million again. We're up to 1 million, okay? So after 20 generations, you have 1 million bacteria, and that happens well within the first day. And so on our Zoom meeting, I'll go back and I'll try to remind you of this little part of my video, and I'll ask you guys a question, which is how long does it take to get 20 generations? How many hours is that? So think about that. Exponential growth will produce invisible bacteria into these unusual colonies, or however they're shaped. And so I want to go back to that plate and analyze the colonies a little bit. So guys, back on our document camera, we can see that all different shapes and sizes of colony growths occur. Some have color. There can be orange, there can be yellow, off-white. Sometimes you'll even find red or pink, and there are even purple bacteria, but they're just not that common. Now, most bacteria you can see are white, and most bacteria are also opaque. They have a milky color to them, but occasionally you'll see bacteria that are clear, translucent. And one thing I would like to tell everyone is, if you do an experiment like this where you sample the environment, you have no idea what you're growing. We could be growing something as dangerous or deadly as Bacillus anthracis. Cloudy colonies that look like this are usually Bacillus species. So it's not a good idea to open these plates. We don't know what these mystery bacteria are. And when they're in their invisible amounts, most of the time they're harmless. But now we're growing these bacteria that could be harmful into large numbers. These are huge numbers that could represent an infectious dose if you were to accidentally come into contact with them. So I'm going to recommend in an experiment like this that we never open the plate. But I do want to flip the plate over so that we can at least look at what the plate looks like, or what the colonies look like from the top side. We do have an issue though, which is that the lid is cloudy. I'm going to go ahead and go into the biological fume hood in the other room and swap this lid out with a clear lid so that you can see this more closely. I'll be right back in a moment, and we'll continue the analysis of these colonies. So here's the plate now with a clear lid so that we can see through it. And I can zoom in even further, and I'll see if turning the light on helps. It does help a little bit. And we can really see these colonies from the top side as they are growing up from the agar nutrient jelly-like substance on the bottom and then growing up towards you. They look like usually little dome-shaped structures. Depends on what species they are and how they grow, but you can see the different colonies. Some colonies are shiny, other colonies are rough and dull looking. Bacillus species typically are rough and dull looking. Bacillus anthracis can be most readily identified by growing these cloudy, rough looking colonies on blood agar. And when you grow them on blood agar, they actually look like fried eggs that can peel up at the edges of a fried egg, like it's in a pan, but it's on blood agar. And if the edges lift off and it has a fried egg appearance, it could very well be a deadly organism like Bacillus anthracis. So again, I'm being very careful, but I wanted to at least take a look and see about all these different organisms. The organisms that are off-white, small, smooth, round, regular in their appearance are usually skin bacteria. There are a lot of skin bacteria, even on the ground, even on the bottom of your shoes, because, I don't know if you've heard this before, but it's estimated that in an environment where people live, that the dust inside of places that are human dwellings or whatnot, that the dust is like 70% skin cells. Oof, it's kind of gross to think about, but those skin cells have bacteria stuck to them, and so dust commonly does carry bacteria, even from that one event, but dirt in general, very contaminated. These bacteria that are irregular in size are typical of soil-based bacteria. This looks like a soil sample, and there's no surprise that with the variety that comes from a soil sample, it would just look just like the bottom of my shoe. Let's compare that to my cell phone. So the cell phone, okay, I'm gonna try to get both in the picture. My cell phone has a surprising number of bacteria, no question. That's not exactly like a good thing in my mind. I mean, yes, I know that we have bacteria all over us. You need to be aware, you're wearing like a suit of normal flora bacteria all over your skin. But your cell phone, or whatever it is that you commonly touch, door handles, keyboards, you name it, we call those objects fomites. And I'll spell that out for you, F-O-M-I-T-E in our lecture. And what you'll see is that when you sample fomites that are common use items, they look just like a skin sample. So if I took a sterile swab and wiped it on my skin and then put it on the plate, which we'll do later in the course, so we'll get a chance to see this kind of result from a straight skin sample, but this is what it's going to look a lot like. This irregular looking colony here, I have no idea what that is. We can take a closer look at it. I don't know how that popped up. Who knows? You know, maybe there was some dirt on my cell phone. I mean, you know, that definitely is possible, right? Let's take a look. I'm kind of just trying to get a better look at it top side. Wow, that's pretty fascinating, you guys. If you look at this colony, it has these strange, I don't know how to just, I'm trying to think of the right term here based on what I'm seeing. When you look at it off camera, just directly, it looks like it's waxy. And these waxy type growths are usually from unusual species of fungi. There's a species called trichoderma that looks like that, and think about it, trichoderma. So it could very well be a fungus that comes from the skin. We have some fungi as normal flora as well, just not nearly as many as the bacteria. So maybe I have some trichoderma on my skin. But the only way I would be able to identify these more certainly would be to put them under a microscope and actually look at their structures. We're going to do that later in the course. For now, though, we're just taking kind of a broad look at this. And speaking of a broad look, you can see all around, situated around this weird waxy one, that there are all these fairly similar looking colonies of bacteria. And those are what staph bacteria look like. All right. One of the staph that we're going to be using today is called Staphylococcus epidermidis. And Staph epidermidis growing in this slant is the most common skin bacterium on all humans. So it's what it is to be human. Just like we all have E. coli in our gut, we all have some Staph epidermidis on our skin. Our book's author for our textbook, Tortura, says that as many as 90% of the bacteria that grow on your skin are just 90% Staph epi. So that's a really important and beneficial bacterium when it's on your skin, as opposed to, say, Staph aureus, which can also be normal flora, but we don't like the Staph aureus because it causes disease in many instances, whereas most of these are probably harmless bacteria that fall into the Staph epidermidis category. All right, that's enough about that. We're going to incubate all of these over the weekend, as I was mentioning, at room temperature, that they're going to be incubated for longer. So I'm going to leave these out for room temperature incubation. We'll check back on them one more time during our next video. And let's move forward. There are many other things to do. Just as a reminder, guys, you can look at the different vocabulary terms. I'm not asking you to memorize them, but even that, like for instance, that waxy one, it was definitely irregular in its colony shape, and I would say that it had irregular to maybe perhaps filamentous kind of appearance, and also waxy, which isn't listed on here. And so you can get these different vocabulary terms. There's a good reference for you guys. Here's an example. Look at this. So here's a picture from your lab manual showing mixed soil sample growth, and it looks quite a bit like the bottom of my shoe, as that sample was. Or commonly students will swap the bottom of their handbag or their purse or something like that, backpack, and it'll look a lot like this. Here are the colonies of Bacillus anthracis, and it's a little hard to see on here, but these do have the so-called fried egg appearance, because there's a center that looks like an egg yolk, and that raised area looks like that. As we see here, it has a really unusual term called umbonate. That's actually my favorite vocabulary term to describe all of these. It's just so unusual, umbonate. But that's what Bacillus anthracis looks like. And this is growing on blood agar. Here are some different varieties of pigments that bacteria can have, or many different microorganisms can have these unusual colors to them. So there you have it. By the way, I mentioned Klebsiella pneumoniae in my lecture earlier, and you can see that Klebsiella pneumoniae, which is one of the causes of pneumonia, fluid buildup in the lungs, has a mucousy, sticky kind of what's called a mucoid surface, and that's because they stick really well to the lungs, and unfortunately that makes them very difficult for our lungs to fight off. There's your immune cells called alveolar macrophages, as we'll learn about phagocytes, as they're also called, will try to ingest and digest these bacteria, and they have a tough time with these sticky mucoid colonies when they're in the lungs. All right, let's move forward. We were looking at microscopy last time, and one thing I wanted to point out, hopefully the white balance on the camera today will be able to show this particular slide better. So if you didn't get a good look at that last video, when I recorded this and wrote these in, I'm hoping that now is showing up better on camera. Just like the previous slide here when I was talking about the letter E being flipped in mirror image reversed. So hopefully you got a chance to see that, or you're getting a chance to see that better now. All right, we're going to continue looking at specimens under the microscope, but before we can do that, we need to generate a new specimen. And one of the specimens that I'm going to have you guys look at is cheek cells. We're going to make a wet mount of cheek cells. So we'll skip back to this. We're going to perform a simple stain on some cells inside of our mouths, or inside of your mouth, by scraping in there to get cheek cells. And before we do that, I want to tell you guys about how we need to add some color. All right, a cheek cell, like most of the cells in your body, aren't going to have really much color to them. They're usually clear, and they're hard to see under the microscope when you have a bright light under the microscope and then you're trying to add some color. So we need to understand a little bit from the lab manual about stain technology. And so the simple stain is the easiest way to do this, to perform this function of getting the cells to take on some color. We're going to use a dye called methylene blue, and it'll stain the cells. They'll soak up the blue color, and you can see a picture of what the molecule looks like. This is actually a molecule of crystal violet, different dye, but regardless, we're going to look at this molecular structure a little bit. It's very similar to methylene blue, and we'll see how this works to colorize cells that we want to study in lab. Let's see now. A stain is said to have two components: a colored molecule that then is dissolved into a solvent. And so when we look at a stain, we call the colored molecule more technically a chromogen. All right, so the chromogen is the colored molecule that makes the stain, but then it usually comes as a powder that you have to buy, and then you measure out a certain amount of this on a scale, and then you mix it into a certain amount of water, and then you get your stain that's functional so that you can soak it, soak the cells. And so usually water is used as the solvent, although alcohols such as ethanol being the safest and most common, or maybe methanol might be appropriate depending on if ethanol doesn't work. So methanol-based stains, which is like wood grain alcohol, that is dangerous to humans. It's a carcinogen. We don't use methanol unless we're like using a fume hood to filter the air and make sure that, or to get rid of that bad air so that you don't breathe it in. For intro to micro labs, we only use ethanol or water-based stains, or water-based solvents. Let's see now. The oxochrome is a technical term that we look at in terms of analyzing what a stain, what a chromogen is like inside of a stain, and it is the charged portion. So if you look closely here, there's a little plus charge there on this molecule, and that gives it then this positive charge that will be absorbed by the cells. And the reason for that is that cells have the opposite charge. All right, so stains typically have a positive charge, and let's see, I'll use this page right here to just draw a little diagram so you guys can remember that a stain, let's give it a color. So I'll change my pen to red or some color. How's that? Make a stain molecule here. And this stain, or chromogen, gen means to give birth to, and chromo means color. This, change colors back, is going to be absorbed by the cell. Now, let's say this is your bacterial cell. Okay? Now, bacterial cells are said to, well, all cells really, are said to have an overall net negative charge. And this is an interesting topic because we have to understand quite a bit about the cell and how it's built. The cell membrane is made of chemical structures, primarily phospholipids. Now, maybe you're familiar with this. If you're not, don't worry. Phospholipid has in it a chemical called phosphate. So to explain, if you've had any chemistry, phosphates have a negative charge. So in chemistry, it's PO4, and then usually there's a minus charge, like PO4 minus 3 is how phosphate is mixed or floats around inside of a solution. So phospholipids, though, which are part fat and part phosphate, are what make up the cell membrane. Also, inside the cell, there are chemicals that carry energy, like ATP, adenosine triphosphate, more phosphate, and phosphates have negative charges. We also have the DNA. Genetic material has a net negative charge, also because of phosphates. And so what we see with cells is that for the various reasons that I just mentioned, there are these negatively charged, there's this negative charge character surrounding the cell, okay, and also inside the cell. Now recall a moment ago that the stain, which has a positively charged chromogen, will be attracted to the negatively charged cell. So opposite charges attract. Let me put a positive sign by the chromogen, right? This thing has a positive charge. So when the chromogen in the stain is mixed with the bacterial cell or whatever cells you're staining, we're going to see these positive charges attack and combine with the negatively charged cellular components. So now your cell is red. You end up with a red cell. Or whatever, maybe I don't need to specify red. Let's say a stained cell, because there are many different colors of stains. A stained cell. And that's how a simple stain works. And if we go back one slide, we can see that there's also a technical term here that you guys should know called the chromophore. Now, chromophore is referring to the part of the colored molecule, the chromogen, that gives rise to the actual color. And so chromophores, it says, chromophore groups, as they're labeled here, are these six-membered rings. So the more six-membered rings there are in the molecular structure, these are called benzene rings. These modified benzene rings, as they're referred to chemically, are going to reflect back bright, vivid colors. The more chromophores there are, the deeper and more intense the color. So we can see here on this molecule of crystal violet that there are 3 chromophores, so it has a really deep, vibrant purple color that your eye sees. And you have the oxychrome that's charged. This entire thing, if I put a circle around it, is the chromogen, containing the chromophores. Let me change back to black here. So containing the chromophores and the charged oxychrome, this entire molecule is the chromogen. This whole thing, chromogen. And that's how simple stains work. Now, some of you have asked, especially early in the semester, should I get the lab manual? Does the lab manual help us, help students to study? I gave you all the information that you need, but this may be the only time that I'll actually go out of my way to say, okay, let's read from the book for a moment. It may not be the best way of teaching to just stand and read to an audience, but I am going to read just a couple, maybe a few sentences, a paragraph out of the lab manual to show you that the way that I teach is very much from the textbooks and the lab manuals, both for accuracy as well as a way for you guys to get a lot out of them. If you do decide to invest the time in reading, I highly recommend that you do the reading in the theory and application sections of each experiment, like the simple stain exercise 3-5. So what I'm going to do is switch over to the document camera and read to you for just a minute or two. Okay, so this is the lab manual. It's entitled Microbiology, Laboratory Theory and Application by Leboff and Pierce. This happens to be the 4th edition, but a 3rd edition will work a-okay. There may just be a little bit of a difference in numbering. And in here, let's look at the simple stain. Okay, so we'll zoom in for just a little bit here, and we'll take a look. All right, hopefully you guys can see this okay. And as I said, I'm going to just read off a little bit about this because it shows how my teaching style works. It says stains are solutions consisting of a solvent, usually water or ethanol, and a colored molecule, often a benzene derivative, and that's your chromogen. And the term chromogen is bold-faced, so you know that's a vocabulary term you should know. And then it says a chromogen may have multiple chromophores, another term that we went over. And so then each chromophore adds more intensity, as I was saying, and then it says the oxychrome is the charged portion of the chromogen colored molecule, and it allows it to act as a dye through ionic or covalent bonds between the chromogen and the cell. We didn't talk about ionic, but that's referring to the charge, and in this case, positive charges are attracted to negative charges, so opposites attract, and the negatively charged cells will absorb the positively charged dye. And here is a figure that shows that. So we can see that the negatively charged cell is absorbing the positively charged dye, and so everything's here for you. I hope you find the book to be valuable in the regard that it does really state and summarize, or even dive more deeply sometimes into the material that I teach about. So you'll learn more. It'll give you a chance to crystallize that knowledge, maybe by seeing it collected into better wording. Me lecturing, I'm not always the most efficient with words. Reading may be a more clear, concise way for you. I know it is for me. I try to speak clearly, and but I don't read like a book. I don't speak like a book reads. Okay, so there are actually 4 dyes that I'd like you to spend time having in your minds as just the 4 basic simple stains. Everybody needs to memorize that all the general microbiology, basic microbiology laboratories around the globe have a crystal violet supply, and methylene blue, and safranin, and malachite green. All of these are so common that you'd find them in practically any lab. One of these, the safranin, is the one that doesn't quite give you an idea what it looks like, but it has a reddish pink color. And so anyhow, simple stains all have the same thing in common. They have an alkaline pH, so if you've heard of like acid, the opposite of acid in chemically speaking is an alkaline substance, also known as a basic substance. So basic or alkaline dyes are able to perform the simple stain, and they carry the positive charge. Okay, now it's time to stain, or to simple stain, some cheek cells. And the end goal is going to be to see something like this. And I'm going to reach into my mouth and scrape out some of those cells, and hopefully I'll get an image as good, or maybe even better, than what we see here. That's what we're going to set up. Now I'm going to get all the equipment and give you guys a close-up look at exactly what it is that's going to be set up here. All right, step 1 says place a drop of methylene blue simple stain onto a slide. So I'll switch over to the document camera to give you guys a close-up view. And I have a slide right here. And I'm going to go ahead and place a drop of methylene blue on it. Before I do that, I'm going to dust the slide off. I just ripped the paper. Okay, I'm going to dust off a slide because sometimes, even though they're brand new, they can be a little bit dusty. And now we'll put the drop of methylene blue on there. Okay, so how much is 1 drop? We've got the little dropper bottle. Loosen up the squeezy bulb part, and 1 drop is going to be a whole drop that you allow gravity to pull out of the dropper. If I touch the dropper down physically onto the slide, what will happen is it'll take out less than a drop, like say a half of a drop. So we want 1 whole drop to do this procedure. So here we go. I'm going to put it right in the center of the slide, but I'm not going to touch down. We'll let gravity do its job. So I have my drop of methylene blue dye, and the second step on that set of instructions was to stir in some cheek cells. And I have a cotton swab. This is one way to get cheek cells. Another way is to use a toothpick. I think that the toothpick actually works a little bit better, but I didn't find any at the current moment. I think what I may do is use the back end of the cotton swab, which is a wooden stick, as a toothpick. All right, so I'll carefully get this out of here. I'm going to put this part against my cheek cells and move it around. And I'm sure that even though, you know, it's not like I drew blood or anything crazy like that, but I did a good scraping that I could feel. Hopefully got some cheek cells. Doesn't have to hurt, but I'm going to move them around in the drop so that they're not just piled all in one place. Okay? So now that I've moved them around, what I'm going to do is put a cover slip on there. So this is our last step, is we'll put just a little cover slip right on top of there, and the dye will spread out nicely and allow us to have a fresh specimen to put under the microscope. Now there are a couple of air bubbles that are on here, not a problem. If we wanted to get every last air bubble out of there, we could use a special little device for fluid handling called a pipette, and we could put the dye under there by pipetting it. But it isn't going to matter, because the microscope is only going to look at one little tiny bit of this, and I'm just going to tell you don't look where the bubbles are. So I'll get the microscope out, and the next thing you guys are going to be seeing is a view of the cheek cells at various magnifications. We'll start with 4x, and then we'll go up to 10, 40, and we're even going to do oil immersion, which is the last part of our lesson in microscopy and how to use the microscope. All right, so now we're looking at a view through the microscope, and this is under the lowest power called scanning lens or scanning power. And all those little objects that you see scattered around that are a little bit larger are mostly cheek cells. At the end of the pointer, which is that black needle device coming from the upper or from the side right, it looks like maybe there's some kind of a hair or something, which doesn't surprise me. I think the slide itself was kind of dirty. I'm going to go ahead and switch to low power. So I'm going to grab a hold of the nose piece and rotate the lens into place. Now recall that it'll click into place, and we should only need to do a little bit of fine focusing, which is this knob here. Because this microscopes are parfocal, we shouldn't need a whole lot of focusing. You know, no coarse focus adjustment, just a little fine focusing. So what I'm doing right now is trying to get this back in focus. Ah, look at that. Okay, so I'm doing some fine focusing, and now you can see the cheek cells at low power. So this is 100x. They still don't look very large or detailed. I can see that what looks like a hair just above the pointer. And anyhow, so we have all these different cheek cells in here. And then now I'm going to go to higher power. You really need a high power lens, like 400x total magnification, or even the highest magnification, which is oil immersion. And so we're going to be looking at this under high power and then oil immersion in just a few moments. So let's go back down here. We don't touch any of the controls. We just simply carefully click the high power lens into place. I'll do a little more fine focusing. See how this turned out. Hopefully something will come into focus. Now we're getting into a more detailed situation here. So here's a cheek cell that turned out pretty well. That was a fairly crude way of biopsying, right? I mean, ripping them out of off of your cheek like I did isn't something that's going to produce all perfect results. But this one's okay. And if you look to the right-hand side
nearest to the pointer, the nucleus is observable. And so the nucleus is kind of to the side of this cell. And now we're going to go to oil immersion, which is the highest power, 1000x. And we'll be able to see these cells in even more detail. But before we do that, I want to show you guys how to apply the immersion oil and understand how the oil works. So we'll come back to this in just a few minutes.
So we've looked at our cheek cells under scanning power, low power, high power, and now we're going to figure out how to use the oil. I'll move the microscope out of the way, and let's look for our lesson about how to use oil. So here is the slide about how to apply oil and what the use is in the oil. Here's a diagram showing the microscope down from the light source at the very bottom, and light comes out of the bottom base of the microscope in a straight line, and then it hits the condenser lens, which is beneath the stage, and the condenser lens then directs the light into a cone of light so that there's a fixed beam or a point that's then focused coming up through the specimen. And the specimen are going to be the cheek cells that we just saw, and then that light is going to go up into the objective lenses or whichever objective lens is in place. But we're going to have to place some oil on there in order to get the highest powered oil immersion lens to work. And the reason is this oil provides many advantages. Let me tell you about the oil. It comes in a little vial that is this clear oil liquid that originally came as an extract from a tree called the balsam tree. And the balsam tree has this very clear oil that has the exact same refractive qualities as glass. So think of this almost like liquid glass. And to demonstrate this, by the way, this is they now have a synthetic version of balsam oil that's technically a little bit better, and but it does the same job. And so I wanted to demonstrate about refractive index. The term index of refraction refers to how much light bends or refracts as it goes through a given substance. So light goes through the air, and air is all around us. It's pretty hard to escape the air, right? So when we think about light going through the air, we know that the air can affect light. We don't really notice it unless we look really far away. So if you imagine like looking at the stars up in the sky late at night, if you can manage to see the stars, if it was a windy night especially, you might start to notice that the stars flicker. And they appear to be flickering, but are stars really turning on and off? Right? I highly doubt that most of the stars that you're seeing light years away, millions or billions or whatever of units away out in outer space, they're not really turning on or off. It's that the light is being disturbed by roughly 62 miles of atmosphere, air in our Earth's atmosphere. And so the air is given a default refraction of 1. So we can write this down. The index of refraction, index of refraction is 1.0 for air. But then, what about when light travels through something thicker, something more dense, like glass? Right? Glass is clear, we know that light will go through it, but it's going to have a different effect on the light. And the index of refraction for glass is 1.5. glass bends the light more. And we can see that, even though the glass is clear, we can see that light bends around it and interacts with it differently. Now, the immersion oil is also the same index of refraction as the glass. And so we refer to the oil as like functioning as if it's liquid glass. And one way to figure this, to see this effect as a very real phenomenon is by holding up the immersion oil, and if you unscrew the lid, there's a little glass rod inside, and it picks up the oil, and you can use that little glass rod to drop a drop of oil onto your slide or your cover slip. I'm going to see if I can demonstrate the effect on the document camera. Let's take a quick look at how this looks in the document camera. So we'll zoom way in on this, and you can see if, let's see, that's not showing it very well. Let's try something else. If I hold this up in front of the camera, you can see the light that comes through it. And when I lift the glass rod out, we can see the glass rod. As the glass rod dips down into the oil, it starts to disappear. So the part of the glass rod that's inside of the oil really isn't able to be seen. And that's because it all has the same index of refraction. So if you look at it as changes of index of refraction, you have light, and then the light hits the glass vial, and it changes from 1.0 to 1.5. Then there's air inside the top part of the vial, so it goes from 1.5 to 1.0. Then it hits the glass rod, 1.5. Then it exits that and goes to more air, 1.0. And then it hits the last part of the vial, goes back to 1.5, and then it travels to my eyes and goes back to 1.0. Every time we change an index of refraction from 1.5 or 1.0 to 1.5, we can see that change happen. But down in the bottom of the vial, you've got the light going 1.0, 1.0, hitting the glass, 1.5, hitting the oil, hitting the glass, hitting the oil, all 1.5, and that's why we don't see the glass rod inside the oil portion down below. Anyway, it's kind of like a little magic trick. Switching back to our slide about oil, a couple of key factors can be realized then about the use of immersion oil. All right, this helps to focus the light through the lens. And what that means is that air might actually escape, and if it escapes out of the sides, what did I just say? Air, I meant to say the light can escape out of the sides through the air. All right, but if you put oil in there, what happens is it seals out the air, and that way it helps to trap the light so that more light goes through the oil and up into the lens. And so essentially you have a sealing out of the air, which not only helps to trap the light, but it also keeps the index of refraction consistent, which means less disturbance, which means a clearer image. So if I was to ask you guys to think of a couple of reasons or describe a couple of reasons why oil is important and necessary to use with the oil immersion lens, we could write down that the oil traps light and the oil increases the index of refraction. And because it's the same as glass, that increases the clarity under the highest magnification. Okay, so those are two big points about oil and why it's necessary. But it's only used for the highest level of magnification. We never use oil on the other three lenses. So going back a few slides, when we look at this table, the only lens that actually gets wet is the oil immersion lens. If you get any of these other lenses wet by accident, you always have to clean them off. And speaking of cleaning lenses, I'll tell you about this. I would tell you about this many times during the semester. We have this lens paper. Lens paper is the proper type of paper that will clean off the lenses if any of them get oily or dirty. And we always clean off our lenses at the end of using the microscope. If there's any liquid on them or fingerprints, definitely immersion oil, we always need to remove it with some lens paper. We never use paper towels or Kim wipes. This is unacceptable because it will scratch lenses on microscopes. So what I'm going to do at this point is apply the oil and show you guys what the cheek cells look like under oil. Hopefully we'll have some good luck with that. The cover slip is floating on top of the cheek cells, and sometimes the cover slip moves around, and so we're going to try hopefully to get real lucky and see them under 1000x magnification. So we'll get some high detail of my cheek cells in just a moment.
All right, so we're back in focus under high power 400x, and I'm going to attempt to put the oil on. So we'll take this away and we'll look back down here. When you apply immersion oil, you have to make room to put the oil on. So I'm rotating the nose piece so that I've got some room to put the immersion oil on. And now I'm going to go ahead and use the glass dropper to apply a drop of immersion oil. So gently, you place the drop of immersion oil on top of the cover slip. It may be a little hard to see here, but I put that oil so that when we rotate the oil immersion lens into place very slowly and carefully, it'll actually physically dip down into the oil and click into place. And now with just a little bit of fine focusing, we should be able to see the cells. I'm using the fine focus. Beautiful. All right, if I do say so about my cheek cells. Okay, we have a cheek cell in focus, and besides the nucleus, which you can see to the right-hand side of the cheek cell, closest to the pointer, there are some other dark objects on there, and those little dark objects, if I get them in better focus, are bacteria. Bacteria are the smallest, darkest little objects stuck to the cheek cell, and that's completely normal. Everybody's going to have some bacteria stuck to their cheek cells. The number of bacteria in your mouth is easily more than all the humans on Earth. You have well over 10 billion bacteria inhabiting your mouth, and the populations go up and down as you brush your teeth and use rinses like mouthwash and floss your teeth and all that. But this is a pretty good example of a cheek cell, and it's been stained with, simple stained with methylene blue. And so there you have it. And take into account how small the bacteria are, those little tiny purple dots or blue dots, and then how big the nucleus and, of course, the whole cheek cell itself is giant compared to the bacteria.
So now we've seen the cheek cell at 1000x maximum magnification, and we'll go back to the picture from the lab manual. And wow, this person had a whole lot of bacteria on their cheek cells, but it made for a nice picture. We really can't judge whether or not that's a healthy mouth or not. I'm just kind of making the comment that there are quite a few little bacteria scattered around, and the nuclei that you see are well centered inside the cheek cells. So these are more model cheek cells compared to what I showed you. But, you know, I did my best, and that's all we can do in many cases, but we got the mileage out of it that we needed to. All right, let's move on. Guys, how about aseptic technique? Aseptic technique is an essential hands-on technique that it may be the biggest reason why you guys take microbiology lab. And so we need to learn these words and then go through the motions of learning how to keep ourselves free of contamination and to not contaminate our equipment or anything around us, any others around us. So what does this word aseptic mean? Aseptic means without contamination. So we need to learn which techniques are effective at preventing contamination. And so we're going to go through steps that are listed in your lab manual, exercise 1-3. And the tools for aseptic technique that we're going to be using straight away for our first exercise are the Bunsen burner, and then to move the bacteria around from a sample tube into a fresh tube, we're going to use an instrument called the loop. All right, this is a yellow plastic loop. They have these single-use disposable loops. This is a metal one that we can use many times by flaming it in the Bunsen burner flame to sterilize it. And so at this point, I'm going to do a dry run, just like you guys would practice. And the dry run is to just show the technique, pretending that we have the Bunsen burner lit and that we're transferring bacteria from one tube to another. Now I want you to keep in mind something before we get started with this dry run, which is that we need to know a little bit about the anatomy of the Bunsen burner and how it functions best. So I will use the previous slide for just a little empty space to outline the parts of the Bunsen burner and why it's important to know that we have it adjusted properly for our exercises. So you have your Bunsen burner standing on a little stand, and at the base of the Bunsen burner barrel there will be a hole, actually multiple holes. There are 4 holes on the Bunsen burner, basically straight ahead, and then at 3 o'clock, 6 o'clock, and 9 o'clock around the Bunsen burner. And those let in air so that it can feed the fire. If you close the Bunsen burner holes by rotating this little metal sleeve that can open or close the holes or partially close them, you're going to start to restrict the fire. We really want the Bunsen burner holes to be open. The only time I know that we close them is if you're having difficulty keeping the flame lit. Occasionally there can be some carbon buildup or something that could cause the holes to not function optimally. So we're going to keep this holes open for air. And when your Bunsen burner is lit, up here at the top where the gas comes out, the flame will have 2 parts to it. The part of the flame that has this central inner triangular sharp cone has to be there. If the air holes are closed, that part goes away and the flame gets taller and softer and actually not as hot. If you have the air holes open and everything adjusted properly, the gas pressure, you don't want to have it too high or too low or it'll blow out. You get all that right and you're going to be looking for this inner cone to be there. And what we're going to be doing is aiming to work at the tip of the inner cone. So always work to flame objects at the tip of the inner cone. Or how, you can see what it is. This is the part that we're going to label as the hottest. And that's where we want to aim. So I'm going to pretend temporarily to do this with the flame not lit, and then afterwards I'm going to go live with the transfer of bacteria from one test tube to the other. All right. So the first step that I'm going to do is flame the loop to sterilize it off, so that it's prepared to pick up bacteria safely, and then I'm going to transfer from one tube to another. Let's see. I'll go ahead and try this out. So let's pretend that the first tube I'm going to be working from is this one with the yellow cap. We'll call this tube A. And we'll say that the bacteria from tube A are going to go into tube B, which happens to have a black screw cap on the top of it. All right, so I take my loop, and the way that you hold the instrument is by the back of the handle. Oftentimes there's a little rubber handle on there to remind you this is where you hold it. You can hold it just like a pen or a pencil, but with a pen or a pencil, you would be down more at the business end to control the tip for writing. In this case, we don't want to do that. We want to put our hand back on the loop, and then we'll grab a hold of tube A. So I've got the loop hand, I've got the tube hand, and then what I'm going to do is carefully flame the loop until it's orangey red hot, and then once that's sterilized, I need to give it a moment to cool. And so what I'll be doing next is focusing on this tube hand. My tube hand is actually grabbing the tube near the bottom of the test tube. We don't want to be too close to the top because we're going to flame that tube before we go into it. So here's how we remove the cap. You take your loop hand and you wrap your pinky around it like that, okay? Now, at this point, I'll roll the open tube over the flame, and then now this loop is nice and cool. I'm gonna go in like this, and I try not to touch the sides on the way in until I get about halfway down in there. And now I can relax. Do a little bit of dirty work, whatever it is that you're picking up your bacteria, right? This gives you a chance to refocus after you pick up the bacteria. Now, focus back in on trying to exit that tube without touching the sides. Once you're out of the tube, we can flame that tube again before we recap it like that, okay? And at this point, we need to be really careful, you guys, because we have a loaded loop. A loaded loop has bacteria on it, and we're going to put it into tube B. And when we go to do that, we now have such a small cap that if you try to wrap your pinky around it, there may be some issues. First of all, we don't want to have the bottom edge of like the palm of your hand, that skin. You don't want your skin wrapping around and touching the bottom edge of the cap. So rather than doing the pinky wrap, which I'm sure if you have tiny, small fingers, you could do the pinky wrap, but this would probably give me I think it'd be like arthritic with this thing, my finger cramping up very quickly if I did that all the time. So I want to show you guys a different way to do that. All right, the way to do small caps like this is you take your loop hand, you put it palm face up so that your palm is facing the ceiling, and then guide the screw cap between the pinky and ring finger, and then unscrew the tube and leave the cap like that. Now look what this accomplished. I now have the cap out of the way, my skin is not touching the bottom edge of the cap, and it's not rolling around on the counter. We never want to put the cap down, but we do want to keep it face down so that contamination from the air can't fall into it, and it's out of the way safely. And before I go into tube B, I'm going to go ahead and roll it in the flame, and then carefully I take my loaded loop and I go down in there without touching the sides, and at this point I can transfer my bacteria in. This gives me a chance to relax again because I need to focus once again to get that loop out of the test tube carefully without touching the sides. Now, let me stop for a moment and point something out to you guys since we're on a dry run. When you're coming out of the test tube, sometimes students are really shaky. They might be your first time and you're nervous about it, and if you have a really shaky hand like this, then you're likely to hit the sides. And if we hit the sides on the way out, you're going to contaminate the sides. And even worse, let's say you're really bad about it, you can fling bacteria. This is like it's spring-loaded. You'll fling bacteria all over the place, and we'll have a bacterial party out of bacterial confetti. Bad idea, guys. So sometimes I'll see students and they're kind of shaking. They say, geez, I don't know how I'm going to get out of here without shaking. And so one other solution I've seen people do, and this is not a good one, they'll just go really fast. Now, I know that the hand is quicker than the eye. The loop is even quicker. The problem is how do we know when you did that just now that you didn't hit the sides and fling bacteria everywhere? That is not a way to get out of there thinking that maybe if you're quicker that you're steadier. So take a deep breath, focus in, and just learn how to be steady enough. Maybe just move the tube hand and leave your loop hand steady. But when you get out, you still want to flame that test tube, whether you touch the sides or not. Just to be on the safe side, we flame that and roll it in the flame for a couple seconds, and then we carefully screw it back in. You only have to screw the test tube on about halfway. Put that down, get it off your hands, and then go ahead and flame your loop again. You always want to flame your loop before you set it back down so that you don't put any contamination on any equipment, okay? So hopefully everybody follows that. It's time to do this for real, all right? So I'm going to set these aside. They were just for practice. And I need one more slant, one more agar, tube of agar like this. This is the one that has the bacteria on it, and I need one more of these to make my transfer. So give me a moment, I'll be right back. So now it's time to go live. We did our dry run, and now we're going to do the real thing. So we're going to follow the steps that are on this instruction list. And so just a little word about labeling, first of all. I'm going to make my own brand new slant of bacteria, just like you guys would. You have to label this. And we're going to use a Sharpie pen, and we're going to write right on the glass. Now, there is a little area for labeling, but I don't prefer that we do that, that we use that, because once you label on the frosted area that's for labeling, the label never really washes off. But we recycle these tubes, we use these tubes many times over, and if someone writes on their E. coli and it's not E. coli anymore, it just gets confusing. So generally, if you're going to reuse slants like this, you just want to write right on the glass where it's easy to scrub off later. I'll show you how this is going to happen here. It says, make a slant culture. By the way, this is the agar that's in here, the food source, was poured in liquid and then allowed to solidify on a slant. So we have more surface area to spread the bacteria around on, just like we see pictured here. We're going to be reaching down into the test tube to the bottom part of the slant, and what will happen is we'll lay the loop down at the very base of where the slant is. You don't stab it down into the jelly or agar. We just set it down at the base of where the slant is and then move it back and forth like a zigzag or a fishtail. All right, and that'll spread the bacteria around on the test tube. So the first step, we're going to be making slants of either Citrobacter frundii, which is an ordinary bacterium that lives in your gut. It's normal flora. It's considered healthy and beneficial to have in your gut. And we're going to also make your lab partner would make a slant of Staphylococcus epidermidis, SE, and I was speaking earlier about how staph epidermidis is one of the most common and most abundant bacteria on the skin. So let's switch over to the document camera for a quick moment so I can show you the labeling. All right, now, it's really zoomed in. Okay, so here's the area for labeling that we're not going to use. All right, we're just going to go onto the bare glass, and it doesn't matter exactly where we do this. I'll just maybe do it right here, and I'm going to put C. Frundii. You could even just make it simpler for yourselves and put, C. Frundii or just CF, and then your initials. So we'll put my initials, D for Derek, B for Boyer. All right, and that's about as much information as we need on here. You could also write the date if you like. I'm going to leave it simply like that for now. And now I'm going to go ahead and make my transfer. So I've got the tube that I just labeled, and I have my bacteria. That's already labeled C. frundii. If you're wondering what do the bacteria look like already grown, I can switch back for just one quick moment. This is the supply test tube that comes from our preparation beforehand. And it's a little hard to see, but if you look on there, there's some moisture there. If you look on there, you can see that there's a zigzag of creamy bacterial growth all over the slant from bottom to top. And so we're going to be recreating that same pattern by grabbing just a small little bit of that. And the amount that you grab is always far less than I think beginning students realize. The amount that I need out of here is not, it's the smallest amount that you can barely, maybe even, perhaps even just visibly see on the loop. Let's take a look. So I'm going to go ahead and light the Bunsen burner first. Where is the lighter? Right here. Okay, when you light a Bunsen burner, just real quick, you want to make sure that you turn the gas on, but only till you can barely hear that it's on. That's the right amount of gas pressure. And then you can adjust accordingly. You don't want it blasting or it'll just blow right out. All right, so I've got the Bunsen burner going. And then now it's time to flame my loop. Oh, when you flame a loop, you always start where the wire meets the handle. And it's a good idea for efficiency and not wasting time to just hold it more vertically rather than passing through it horizontally. That was a lot quicker to do it that way. And then now I'm going to grab the tube that has the bacteria in it. I've already pre-loosened the cap so that it's easy for me to put my palm face up, grab that cap between my pinky and ring finger, and then unscrew the tube. Now what do? Remember, the loop is already cooling off. We don't want to be in too much of a hurry or we'll fry the bacteria. That would be bad. And then I'll roll the tube in the flame. And then very carefully, I'm going to go in, not touching the sides. I'm going to go down into the slant and just wipe it through a very small part of that zigzag. I have just the tiniest little visible amount. You can't see it from here or on camera, but it's just a little dot of bacteria, not big gobs, just the smallest amount you can see. And that's still millions of bacteria, guys. So we have the cap pre-loosened on the new auger slant. I'll grab that cap carefully. Now I'm flaming it. You always flame tubes before and after entering them and exiting. Okay, so I'm going to go down to the bottom, set it down. There's a little bit of moisture down there, but that's sterile water. I'm not going to worry about it. I'm just going to move the loop back and forth to recreate that same zigzag that you saw on the supply auger. Okay, careful on the way out, I didn't touch the sides. Flame it again, recap it, always just screw it on halfway. We never incubate the test tubes with the caps all the way tightened because the bacteria can run out of air. They can actually use up the air. So I always flame my loop before I can set it back down, and then I'm all set. So at this point, I'm going to place these into the incubator, and we'll move on with other parts of our lesson. For now, that's aseptic technique, you guys, and we're going to learn a lot more about aseptic technique and how to use it on plates and other microbiological equipment where we see what the do's and don'ts are to make sure we're always free of contamination. And so there's that. Okay, good enough. Finally, this is our last topic for today, and it's the negative stain. The negative stain is a lot like the simple stain. So we're going to learn some of the same concepts, but the negative stain is sort of the opposite of the simple stain, which is strange, but if you know the simple stain really well and you've studied to remember the different take-home points about the simple stain, the negative stain will just be like reversing it. And so let's look at this technique. The negative stain, I'll give you an example on reversal. The simple stain, the blue dye that we use, the methylene blue for my cheek cells, it gets absorbed directly by the bacteria, and the slide is clear, but the cells have the color. In the case of the negative stain, we see that the background, the actual glass of the slide, has color, but the little clear objects that you see under the microscope are the bacteria. Now, not every single little tiny clear object in here is bacteria. Some of these can be like oil bubbles or air bubbles or dust, debris, who knows. But the objects that look fairly regular in shape, like these two rod-shaped bacteria connected together. This is a rod-shaped bacterium. These are rods. You may find some little coccus spherical-shaped bacteria as well that are not just simply specks of dust. The things that are not bacteria, that are junk or whatever, are referred to as artifacts. Artifacts appear in slides all the time, and we want to be able to look for the actual bacteria and not mistake them or mistake the dust and things like that, the artifacts, for bacteria. Now, the negative stain has its place in microbiology in that it is a heat-free way to stain the cells. Now, earlier I did a simple stain by floating the bacteria, soaking my cheek cells actually in the blue dye. But oftentimes when we do simple stains, we'll see the next time we do a simple stain, next lab period, that simple stains oftentimes involve heating up the cell samples in a liquid solution so as to dry them out and then apply the dye. The simple stain is different than the negative stain. The negative stain is a way of avoiding fixing the bacteria with heat and drying them out. We do an air drying process because there are some bacteria that are so sensitive to heat that they would either shrink up or break apart if you were to use any kind of heat. And so we're going to do a heat-free procedure here called the negative stain. So let's emphasize that point. The reason that you're doing this is that it's heat-free. Heat-free. And this is important because of the fragile cell types that I was describing that could break apart. One such very delicate, fragile type of bacteria bacterium is the spirochete. And so spirochetes are coiled, spring-shaped bacteria, and the negative stain is one of the only staining techniques that allows us, through ordinary use of the microscope, to see spirochetes. So let me just move forward for a moment. Here's a spirillum, or spiral-shaped bacterium similar to a spirochete. And here we can actually see the spirochetes on a negative stain background. So you can see the negative stain in this case is a really dark ink. It's called India ink. And that particular dark negative stain blacks out the background, and then you can see these little curly, coiled, spring-shaped organisms. And these spirochetes are really important to be able to identify. And that's because spirochetes are known to cause a lot of severe, if not deadly, human diseases. One such disease that I'd like to highlight that's arguably the most important of the spirochete-based diseases is syphilis. So syphilis is a sexually transmitted infection, STI, sometimes referred to as STD, sexually transmitted disease. Whether you show the symptoms of disease or know you have a disease process going on, the infection status is that it's somewhere in the body. Most likely it starts out in a sexual encounter or sexual organ exposure. So unprotected sex, these bacteria are causing mucous membranes and surface skin to turn into an ulcer. That particular ulcer, like at the end here of this picture, this penis, is it's called a chancre. So sounds horrible, and I imagine it must be very horrible. Interestingly, people who experience or develop these chancres say that it's not really that painful, and that's surprising considering how nerve rich the sexual organs are. And the theory behind this, I think reality is that when a person develops a chancre, what's happening is that the nerves underneath and in the skin are being destroyed by the bacteria. So that's kind of a scary reason not to feel it, is that the chancre is causing a sore to develop that doesn't have a lot of pain associated with it. And this is the primary stage of syphilis. So following an unprotected sexual encounter, no condom or way to block contact between genital areas, the chancre may develop. Now, that doesn't mean that oral sex couldn't be a possibility as well. So we can think of sex as just a straight interaction, male, female, and everything, but really any type of oral sex, the throat, the area up in your mouth and throat could also come into contact with the spirochetes. The genus and species, by the way, is called Treponema pallidum. So if Treponema pallidum gets in the throat or some part of the oral cavity, the chancre could develop as well. And any kind of mucous membranes, really, if you think about, another possibility would be anal sex. So none of these are good good facts about the fact that syphilis can get involved in these areas. And if it's fairly painless, you have to think about it from a male versus female perspective to some extent. All a guy has to do is look down and see that and say, oh geez, something's wrong. And if you don't know better, if wishful thinking is all you do, like thoughts and prayers, that's a bad move, because surprisingly, this will eventually heal itself up. But it doesn't mean it's gone away. That's the surprising part. This can get as large as like a 50-cent piece, a silver dollar. I mean, it's giant. But then eventually it heals back up. But what happens is these coil-shaped bacteria are actually diving deeper into the tissue, and they can get into the blood, and they can then bring about a secondary stage of syphilis. This is a rash from basically secondary stage of syphilis where blood vessels are affected. And so sometimes people don't know they have syphilis until they get to the secondary rash that often appears on the soles of the feet and the palms of the hands and other body parts though as well. So we have the secondary stage of syphilis represented by this rash. And this is really too bad if you think about it, to get to this stage before you figure out that syphilis is what's going on. And I say that because guys can figure this out if they go to the doctor and they're smart about it. They say, you know, I see something's wrong here, and the doc could do a sampling and like swab that area, look for the spirochete-shaped bacteria underneath the microscope and make the connection. Okay, if you do a negative stain, you'll see that. A simple stain will not do the trick. We have to use this technique that we're going to learn. But what if you're a lady? What if the area, that the sexual organ, right, up inside the vaginal area, you're not going to maybe feel that there's a chancre or chancres, but then that's going to come and go, and if you don't go to the doctor because you have no reason to think you need to go, then you're already going to be on your way to the secondary stage. Now at this point, hopefully the doctor has the judgment to say, we need to test your blood, we want to make sure there are no bacteria that are causing infection, that there are actually antibody-based tests. There's one called the RPR, Rapid Plasma Reagent, and they will do that on a blood test to see whether or not you have antibodies to Treponema pallidum. There are other tests as well that can be used, but the RPR blood test is usually what they use. And that's the one also that they'll test for before you get a marriage certificate, at least in the state of California. You get an HIV test for like AIDS. You can get this test to test to make sure syphilis is not at work in your body. And in the absolute worst case scenarios, in the end stages, if nothing happens to cure syphilis, people will progress to a tertiary stage of syphilis. And this type of syphilis can lead to deadly consequences. And syphilis doesn't cure itself. So people eventually can experience a premature, pretty torturous death. By the way, the picture you're seeing are what are called gumas. G-U-M-M-A-S. And these gumas look almost like pepperoni pizza. That's awful. At this point, think about what's going on. Syphilis hooks into the skin here during surface contact, during sexual activity, and then you get down the line to where it's in the blood doing blood vessel damage and causing a rash, but eventually now these things are erupting, causing skin eruptions from the inside, breaking back out. Gumas, I just remember the name because the skin looks gummy. Excuse the description, but it's pretty bad. Now, this stuff can also get to the brain. Spirochetes can corkscrew their way through tissues and into the brain. And if it gets into the brain, one of the first symptoms is that it can affect the eyes as well, and it causes an inflammation of the iris called iritis. The colored part of the eye becomes really photosensitive and photophobic, which means light fearing. Looking at light can be really painful. The brain can start to deteriorate and malfunction, and people have died of like dementia, where they're out of touch with reality, and it's syphilis-based tertiary end stages of syphilis. So now that you know why this disease is important, and spirochetes are important to understand, let's also just touch on one other major spirochete-based disease. And if any of you have heard of Lyme disease is not caused through sexual contact. It's not transmitted from people having syphilis. In this case, it actually comes from just taking a walk in the woods, typically. Imagine being somewhere, maybe you're on a camping vacation or you're a hunter or something, you're out, out in the wild. And wild animals like deer, for instance, they get bitten by this little spotted tick, or actually that one's not spotted, but there are many different species of ticks that can carry Lyme disease, which is a bacterium that goes by the name Borrelia burgdorferi. And so take note of these names. You need to know the name Treponema pallidum and that it causes syphilis. You need to know the name of Borrelia burgdorferi and that it's the cause of so-called Lyme disease. Lyme disease causes an interesting rash called a bullseye rash, and it's present in most cases following a tick bite. The tick is always what transmits Lyme disease, but sometimes the bullseye rash doesn't appear like that, and so it can be confusing. People don't always know that they've picked up Lyme disease following a tick bite. But if you ever get bitten by a tick, it might be wise to seek testing to make sure that you don't develop Lyme disease. Lyme disease is one of the most confusing types of disease to misdiagnose, to not diagnose. Doctors sometimes fail to figure out that people have been suffering from Lyme disease for months and in many cases even years. And the Lyme disease is devastating because these little spirochetes can get into the heart, and they can get into the brain and the eyes and different organs throughout the body, and they corkscrew, they burrow their way deep into tissues, and even antibiotics make it difficult to treat Lyme disease. Back on syphilis for a quick moment, you don't necessarily have to, suffer syphilis for the rest of till the end stages, right? Syphilis can be cured by antibiotics. There's an antibiotic that treats this called bacillin, and I'll talk more about it during the antibiotics chapter. But bacillin is sort of a variation on penicillin, all right? It has a similar mode of activity, and right now bacillin is hard to get. We were having a drug shortage, at least as of a couple of years ago when I was reading in the newspapers about syphilis making a comeback. Maybe medical insurance is a problem. Maybe states aren't testing for syphilis. A lot of people aren't applying for marriage and getting marriage licenses because it costs money to get married. I mean, I can go off on a whole tangent about how there are reasons why people are getting these diseases which we hadn't seen in a long time. There's no vaccine for this, but testing and treatment has always been a key. Here again, testing and treatment earlier rather than later, before the body has been damaged to some great degree. The Lyme disease is something that is very difficult to treat in its later stages because the organisms are deep in the body, and a person can go through months, if not more than a year, of treatment before they can hopefully clear themselves of Lyme disease. And even then the doctors suspect, well, is it really gone? Could it still be present, buried somewhere inside the person's body? All right, so now you know a bit about Lyme disease, and tick bites are the key there with Lyme disease. Now, to finish things off, I want to tell you a little bit more about how the negative stain was, as I was saying, was opposite to the simple stain. But before we do that, I want to take a moment and actually set up the negative stain. So let's see the steps. All right, here are the steps to the negative stain. And the reason I want to set it up first is because it takes about 5 to, better yet, about 10 minutes, sorry, 10 minutes to get this negative stain to air dry without the use of heat. And since that's a long process, we want to get that set up, and then we'll talk about the rest of what happens with the negative stain. So let's just look at the steps really quickly, and then I'll put it on the document. Well, instead of the document camera, I'll do a close-up with the camera. All right, so I suppose I'll do that right now. Start back up here with a close-up of the negative staining procedure. So first I've done step one before we get to the close-up that you need to see. Step one was to label the slide negative stain. And now that I have the label, it says that I need to obtain some bacteria from the gum line of my mouth. So I'm going to be digging between where my teeth meet, because occasionally spirochetes can be found there in the human mouth. It doesn't mean that you have syphilis or Lyme disease. The first time I taught this, that was one of the unfortunate things I forgot to mention. And one of my students found some spirochetes from between their teeth. It does not mean you have a disease. It could be normal flora for some people. Not everyone is guaranteed going to find these spirochetes, but when the student found those, oh my gosh, that person was so scared they stayed till the class was over, and they're very quiet, and then they finally pointed out what the problem was, and what a scare. Anyway, unnecessary scare, guys. We're not looking for anything terrible. We just, maybe we'll get lucky and find a spirochete. We're going to do so by digging in between our teeth and applying a half drop of something called Congo Red. So I'm going to go ahead. How do I get a half a drop, first of all? If you recall, I said a whole drop last time we got some dye when we did the methylene blue, was we let gravity do the job of pulling a whole drop of dye out of the dropper. In this case, we're going to actually take the dropper and touch it down, physically touch it, to wick off a half of a drop on the far end of the slide. So I'm going to go ahead and do that. Okay, I just got a little half drop of Congo Red. Oops, Congo Red. And now what I'm going to do is use this sterile wooden swab as a toothpick. I have not been able to find the toothpicks, but I promise this will do the job as well. So here I am, digging between where my teeth meet, up the gum line. Not the most pleasant thing. It's like flossing your teeth. I mean, but you got to do it. You know, got to get the bacteria. I probably got a little lunch out of there too. Who knows? Okay, now I'm going to go ahead and show a close-up of how I mix this in and how I follow the rest of the steps to the negative stain, to making a negative stain. Okay, so we'll switch over to the document camera. Let's get a good close-up of that so you can see that I have it labeled negative stain. I put my half a drop of Congo Red. It looks like more than half a drop, but I think it'll be the right amount. And what I'm going to be using is my toothpick, or in this case, the sterile end of a wooden stick on a swab, and stir that in. Again, we want to do a good job stirring because we don't want the bacteria to be heaped into one place. I'm going to take a second slide and use it to make a smear. That's how a negative stain is made, is by taking this second slide. Now notice the second slide carefully. It actually has cut corners. If you look at what's present here, right at the corner edge, you can see that there's a cut-off corner, and it's on both sides. And those cut-off corners, called a bevel edge, is a special slide that will make it so that the dye doesn't spread all the way to the edges. If you're using this on clinical specimens, you don't want those bacteria at the edges where your fingers are going to handle the slide. So use a bevel edge slide, and you set the bevel edge down in front of the drop of dye, and you back it back up into the dye so that the stain will spread across the bottom edge of the bevel edge slide, and then you reverse it right across as you feather it across to make a smear. Now, I was a little shaky, and I did get some towards the bottom edge of the slide here. But we're gonna let this air dry, and they're harmless oral bacteria, so I'm gonna just say that this is gonna be usable, all right? Now, the bevel edge slide, I'm just gonna put into the biohazard area to get rid of it, basically. We would treat any slides from clinical specimens as not something we just throw in the trash, but something that we would put into the autoclave pile biohazard container so that eventually it would get destroyed without there being any potential infectious material. In this case, realize as well that these bacteria that are mixed around in the dye, they're actually still alive. Simple stains, by contrast, the bacteria soak up the dye, or the cheek cells soak up the dye, and when they do, they die. That's an interesting fact about simple stains, is that the simple stain kills the bacteria or the cheek cells. In this case, what we're doing is we're letting this air dry, but realize that the cells repel the dye. So these are the steps we just followed, but when we look at these cells, the bacterial cells are not soaking up the negative stain dye, and they're still alive until they gradually dry out there on the slide. Now, if we take a look back, we need to take into account one last little lesson while that's air drying. And that's that there are 4 major negative stains, just like there are 4 major simple stains. The 4 major negative stains are Congo red, the one that I just used. It's a light red dye. There's nigrosin, which is a black dye that has a little bit of a grayish purple, lighter black color. This picture taken from your lab manual is a picture of nigrosin used as a negative stain. And then there's India ink. That's the one that I was talking about here. That's just like pitch black. You get the midnight effect here. And then there's also eosin. Eosin happens to be a red dye. Some of you who have taken your like anatomy class, if you've had anatomy, you learn about so-called eosinophils. And we'll learn about those at the end of our course during immunology. But eosinophils are basically immune cells, white blood cells in your blood that when they're stained, when we do red blood cell staining, or I should say human blood staining, you can find that, that the eosin is the dye that actually makes eosinophils red. Now, guys, what is it about negative stains that are opposite to simple stains? So, do we have okay, I don't have a diagram. Let's just make a little diagram right here, understanding that these dyes all have the same thing in common. They are negative in charge rather than positive, like the simple stain, and they are all acidic as opposed to alkaline. So they're opposite the simple stain in that they have the opposite charge and the opposite pH, making them acidic. So when we look at a simple stain, I'll go to a different color here, maybe red again, for Congo red. Here's your color of your dye, okay? And this is a negative stain. And here's an easy way to memorize this if you're ever confused about simple stain versus negative stain. A negative stain has which type of charge? Negative charge. So negative stains or negative dyes have a negative charge. And then when you combine the dye with whatever cells you're mixing it with, this is your cell, whether it's I don't care if it's a gum cell or a bacterial cell or a cheek cell, whatever. Remember, all those different cells are going to be surrounded and filled with an overall net negative charge. And the negative charge is going to interact with the dye. But this time, instead of having opposite charges attracting, we have like charges. And like charges don't attract and combine, they actually repel each other. Like when you turn magnets around the wrong way and the magnets don't want to stick together, they actually like repel one another. And in this case, the negatively charged dye is going to bounce off of the cell and not be absorbed by the cell. And so you're going to see these positive charges all around, but not inside the bacterial cell. So we end up seeing sort of a background of color everywhere where the cell repelled the dye. And there's your negative stain, okay? So it's opposite how simple stains work. That's as much as I think we need to cover today. That answers that question right there. Why is the background stained in a negative stain? The dye is repelled, it's negatively charged, it's acidic. And next time, guys, we're going to be going over the capsule stain. And if you look at the syllabus, be prepared for a video covering all the topics that are listed in day 3. Okay? I'm going to say enough's enough today. That was a long lab session, and I hope you guys have thought about the questions that we'll be addressing during our Zoom meeting. Thanks, guys. We'll see you next time.